Wonderful. And next up we have Dr. Jonathan Glass, who is the Professor of Neurology from Emory ALS Clinic, and he's going to speak to us about stem cells, the hope and the hype. So thanks again for coming.
Thank you for inviting me. This is actually an honor to be here. I appreciate that. And also, it's great to follow Jeremy's initial talk because I think he gave you a very nice overview of the clinical trial process and how important it is to do the science. The answer here is going to be in the science. It's not going to be in press releases. It's not going to be in hope is important. I'm not going to argue that. But it's not going to be in things that we don't actually test in the clinic. And so the title of my talk, which was given to me, but I'm happy to do it, The Hope and the Hype, is about that kind of juxtaposition of our needing to have the hope, but being very careful about the hype. And I'm just going to start here. This is the hope of stem cells. Stem cells are magic. We don't know why they're magic, but they're magic because they're stem cells. Because we really don't know what they do, and it's really hard to test what they do in humans, and so we can think anything we want. So we get cartoons like this, where stem cells are going to help with Alzheimer's disease, Parkinson's disease, spinal cord injury, all kinds of maladies that we don't have any good treatments for, and one of those is ALS. Turns out it's not on this particular cartoon. But in fact, I get letters all the time. I get emails all the time. Dr. Glass, you're my only hope. But I don't think that's true because I don't really know if stem cells work yet. I'm going to give you all of the hope that we have about stem cells. And I am a big supporter of this. I've done a lot of work in this area. And so, but we need to be very true to what we're doing and stick with the science. So when people say, you're my only hope, I said, no, I'm not your only hope. There are many hopes out there, and Dr. Scheffner just gave you many other hopes as well. But we need to test stem cells before we actually move forward and provide it to people. So the idea of stem cells has been around for neurodegenerative diseases have been around not for a short period of time, for actually a long period of time. And so these kinds of review articles have come out at least for close to 15 years, maybe closer to 20. And in fact, we're still talking about this. And in fact, you can see we have the same authors that are still thinking about these kinds of things, using stem cells for neurodegenerative diseases. And so there's been a lot of thought behind this, but there's been a lot of science behind it. There's been a lot of testing behind it. And we're finally moving forward toward human clinical trials. So why would we use stem cells? What's the idea behind stem cells? We've just heard some great biology behind some of the other types of treatments that are being tried, whether they be genetic treatments or inflammatory treatments, or even treatments that try to make muscles move better. But why would we use stem cells? What could they possibly do? Well, stem cells are kind of interesting characters when you study them in the laboratory. We can actually put stem cells in and they can replace cells. Cells that have died. Now, if you think about that in ALS, that might be a difficult task. If we look at stem cells, or at least motor neurons, that have died in the brain or in the spinal cord, it might be very, very difficult to reconnect those motor neurons if we replace them to the muscles that they need to get to. That can be over long distances. And so that might not be the best thing. But there are other possibilities. Something we call trophic support. I call them nursing cells. The idea that these stem cells can get in and surround or interact with the motor neurons that are sick and keep them alive for so much longer. In fact, potentially make them stay alive and reconnect with muscles that they have disconnected from previously. And the third idea gets back to the idea of the immune system, immunomodulation. Stem cells have very interesting characteristics in terms of how they can affect the immune system. They can secrete different types of growth factors and anti-inflammatory factors, and they can reduce some of the damage, at least in models, that happens in models of neurodegenerative diseases. So there is hope here. The idea of using stem cells is not a crazy idea. There are actually some good ideas behind it. And in fact, stem cells have been used in many different types of neurodegenerative diseases in laboratories all over the world. So the other hope is that we're not the only ones thinking about this. There are investigators in other fields who are studying other types of diseases that are actually testing stem cells as well. And the hope behind that is that we're going to learn from each other. And we're going to see what happens in other diseases and use what they've learned in the ALS field as well. And they'll do the same thing with us. So there's hope. And so we've moved forward. And some of the hope comes from animal studies. And as Dr. Scheffner's already told you, when you come up with an idea, usually you have to create an animal model and test your idea in animal models. And so that has been done in many stem cell trials, and this is just one from the neural stem trial. And here what you can see is this is the SOD1 rat. This is a rat that carries the human mutation for superoxide dismutase 1, which is one of the more common mutations that causes ALS in people. And when these rats age, they actually develop a disease that looks very much like ALS. They get weak. And they start to drag their legs, and they eventually die. And so these stem cells, these are neural stem cells that come from this company called Neuralstem, were injected into the spinal cords of these SOD1 rats. And what you can see here, the control graph represents cells that were killed before they were put into the spinal cord versus the live cells here. And what you can see here is in the control graphs, these are the motor neuron numbers in the live graph versus the control graph. And you can see here very obviously that the motor neurons tend to live longer when you use these stem cells. You put human stem cells into a rat. And so that's a lot of hope, and that's actually a very nice trial. And it's been repeated several times, and these animals actually live longer as well. What's not quite clear is why they live longer when you're injecting cells into the spinal cord, but be that as it may, they do live longer. And so from there, it went to large animal validation. And so in order to actually get these stem cells in, humans are not like rats. It turns out that pigs have an anatomy, a spinal cord anatomy, that is very, very similar to human anatomy. And so the idea and the technique of getting these stem cells where we need to get them was all tried in these things called Göttingen mini pigs. And most of this work was done by my colleague, Dr. Nick Boulis. And so he had to develop a platform in order to figure out how to actually target these stem cells into the spinal cord. And you can see here, one of these pigs, you can see their scar back up here from where they had surgery. They do, over the first several days, have a problem, and probably it's related to the pain associated with surgery. But very, very quickly, they come back to normal. And this has been done probably more than 150 times at this point, and the results are very consistent. And so we now have a technique of delivering the stem cells that seems to be relatively safe in an animal model that is relevant to human anatomy. And so again, that's the hope that we can move forward with this. And so we did move forward to humans. We've now done this in 30 people. And you can see here, this is pretty delicate surgery. And so again, you never want to make anybody worse. ALS is a terrible disease, which many of you out there know better than I do. But in fact, we don't want to make it worse. And so what we're doing here, Dr. Boulis is injecting these stem cells directly into the spinal cord. You can see all the blood vessels on that spinal cord, and if you hit one of those blood vessels, you can get into trouble. And this, we put in about a few drops of stem cells that have up to 16 million cells in there, and they are targeted toward the motor neurons. And this takes, in the operating room, about 4 to 5 hours. And we can just do it over a small segment of the spinal cord. But again, moving forward. Can we do it? And then if we can do it, then the question is, should we do it? And the only reason we should do it is if it actually slows the disease down or makes people better. And so we published this work of the 30 patients in several publications. This is just 3 of them. And in fact, the most recent one came out earlier this year, kind of going through our phase 1 and phase 2 trials. Phase 1 trial, as Dr. Scheffner told you, is the first thing is to see if you can do it. And the phase 2, at least in our case, was to see what dose we could get to. And so we increased the dose, and we increased the dose by the number of injections in each patient. We got up to 40 injections into the spinal cord, and up to 16 million cells into the spinal cord. And what this paper suggests is that we can do that relatively safely. And then the next question is, what happens to these cells? And if we put the cells in and they just immediately die and go away, that might not be terribly useful to patients with ALS. And at least at this point, and this will change hopefully in the future, but at this point, the only way to find the cells that you put in is when the patients die, and at autopsy we need to go find those cells. Well, putting human cells into a pig and finding them again is not a very difficult pathological task, because pig cells are very different than human cells, and we can actually mark a pig cell very clearly and mark a human cell very clearly and see where the differences are. And that was all done in the preclinical work in pigs, so we showed that these cells could survive, and that's how Dr. Boulis figured out how to target them directly to the motor neurons. But in humans, it's a little bit more difficult. You're taking human cells and you're putting them into a human, and so you don't have that differential. And so we actually did this by looking for cells. All the cells that we put in, at least in this trial, were from a male fetus. And so, oops. All the cells that we put in were from a male fetus, and we had a female patient that died. And what we can do is use something called fluorescent in situ hybridization, which can separate out an X chromosome from a Y chromosome. And you can see what it does here. The X chromosomes are red, the Y chromosomes are green. And so in this woman who died with ALS but had had a transplant of our male stem cells, you can see that most of the cells are 2 X's. Showing that those are the cells from the recipient, from the patient with ALS. But there are many cells in here that actually have both red and green. And so this woman died more than a year after surgery, and we're still finding these stem cells in that region of transplant a year later. And so these have to be the donor cells. And so that's encouraging, and that's the hope, is that not only can we put the cells in there and put them in there relatively safely, but in fact we can find them a year later, which means that they don't die right away, and they're probably doing something in there. We are now working to identify what these cells have become. Many of them have become neurons, in fact, and we call those interneurons, and we're looking to see if they're connecting with the motor neurons as well, which was already done in the rat studies, but we need to prove that can be done in humans. We're not the only ones doing this. There are other groups around the world who are doing this very, very carefully. And this is the group from Italy who are using what are called mesenchymal stromal cells, and they've transplanted them into patients with ALS into the spinal cord, and they have reported their results in peer-reviewed publications as well. They followed some patients up to 9 years and have been looking at those patients. So we're not the only ones who are doing it. There's another group in Spain who are using blood-derived stem cells, and they're putting them into the spinal cord as well, and they're actually finding at autopsy that these cells are interacting with the motor neurons as well. So that's all hope. I think that's all good, that we're doing this in a very structured, scientifically sound way. We're moving forward before we can claim that anything actually works. So we've reached several hurdles. We've gone over several hurdles, which is, number one, we have an idea that it might work from a biological sense. We've shown that it can work in an animal, and now we've actually shown that we can safely provide this to humans. The next question is, does it work? So that's the hope part. Now I'm going to talk about the hype, which is dangerous. This came from a colleague of mine who was wandering through the city of Atlanta and saw this. Ask about stem cell therapy for your pet. So stem cells are out there, and everybody's thinking about them, and a lot of people are selling them. And they're selling them not only to humans, but here you can see that they're even selling them to animals. And people are buying. People are buying. So all you need to do is go onto the internet, and you will find probably close to a dozen, maybe more, of these types of sites of claims of companies mostly, sometimes physicians, sometimes surgeons, sometimes non-physicians, selling stem cell therapies for patients with all types of diseases, ALS being one of them. There have been some pretty spectacular claims out there. There are patient stories out there that you can read about. These patients are patients who have gone to these places and said they have been cured of their ALS because they have had these stem cell treatments. I can tell you that the ALS community has looked for these people. We can't find them. We can't find that they actually had ALS, number one, and number two, we can't find that they got better. And so, but there are dozens of testimonials out there from all these websites about people who have gotten better. But it's very difficult to actually prove that. And you can see here that, in fact, these places are not singular because they're competing with each other. So the typical treatment is Save Stem Cells, okay? But in fact, this place, which is called Global Stem Cells Incorporated, has the very best stem cell treatments. I may be making fun of this, but in fact, it's not funny. It's potentially criminal. Here's another one, Stem Cells of America. The power of stem cells. Everybody wants to believe this. And they have videos, they have these people working in their what look like very sophisticated laboratories. Again, I can't tell you that they don't exist, but I would be suspicious that these are real photographs. There are no known negative side effects. It's absolutely painless, but you better act now because limited space is available. And so again, they're selling these types of treatments to people who are really looking for answers. And maybe it's our fault that we haven't come up with the answers yet, and that's what drives an industry like this. But it's out there, and I think it's potentially dangerous. But of course, they put their disclaimer in that results may significantly vary, and positive results may not always be achieved. Here's a good one in China. Not only does this one treat ALS, okay, but it treats all these different diseases, including Charcot-Marie tooth disease, a genetically based disease, autism, epilepsy, eye disorders. It's also anti-aging, so anybody who doesn't want to get older can go get these stem cells as well. And they have a separate group called the Parkinson Center. So these types of groups have been investigated. They've been investigated by the United States FDA. They've been investigated in Europe, and many of them have been closed down. And most of them will be in countries that have not been able to close them down yet. Hopefully they will. And the other issue is that if anybody who is providing you with a, quote, experimental treatment asks you to pay for it, you should be very suspicious that that's not a real clinical trial either. So 60 Minutes, which is a long-standing kind of news magazine program in the United States, put together this. It's a multi-part series. They called it Stem Cell Snake Oil. And this is a very interesting expose of a group that would recruit patients in the Western United States and send them to Mexico to have their stem cell therapies. And in fact, they investigated these folks fairly carefully and found out that the whole thing was a scam. They not only took these people's money, but many of them actually never actually got their quote stem cells either. So again, the newspapers, magazines, not only the scientific community has caught on to these kinds of things and is really trying to educate the public about what's going on here. There's a group that's run by a colleague of ours named Rick Bedlack at Duke University in North Carolina in the United States, who has developed this thing called ALS Untangled. Many of us are members of ALS Untangled, and what Rick does is he investigates these things in a scientific manner. So there will be claims made from places and people all over the world about treatments for ALS, and the group investigates. Rick takes the lead on most of these things, although there are other physicians and researchers around the world who will take the lead on some of them, and we publish articles. And the articles actually say, okay, here's the claim. What's the proof? What's the science behind it? What's the biological basis for it? Is there anything to suggest that this might be useful? And so for anybody who's interested in any of the, not just stem cells, but any of the claims that are made for treatments of ALS, this is a good place to go. It is published, I think, for free in the ALS Journal. Is that right? So anybody can get it. And so I would encourage any of you or any of your friends to go here first when you hear about something that sounds like magic. Let's see if we've actually looked into it. We haven't found many that actually seem to meet any kind of scientific criteria for truth. So that kind of hype is kind of, I would argue, is the criminal hype. It's the hype out there from organizations and people who are looking really, I think, to make money. But there's a more subtle type of hype, and I think Jeremy's already brought this up with the idea of press releases. Press releases are just that. They are press releases. They're encouraging, they provide hope, but they're not science. They're not the result. They're not the result of the clinical trial. And so we need to see these published clinical trials in order to really evaluate it. So this is actually our trial. This is the most recent trial from our stem cell trial. And this goes through the phase 1 and phase 2 portions of it. And you can see in this published publication in Neurology, this provides class 4 evidence for that for patients with ALS, spinal cord transplantation of human stem cells can be safely accomplished and does not accelerate the progression of disease. This study lacks the precision to exclude important benefit or safety issues. Okay? So that's the truth. That's what I believe that. That's what I wrote. Okay? And in fact, if you look at the trajectory, this is this ALS FRS-R that Dr. Scheffner talked to you about, and the forced vital capacity of breathing, you can see that of our patients, and again, we only had 30 patients. It's not very many for a clinical trial. You can see there's a lot of variability. This is the trajectory of disease. Some people have very rapid trajectories. Other people have very slow trajectories. We had one patient who actually seemed to get better, and you can see that here for both the ALS, FRSR, and the FPC. Okay, that's the reality of what happened, and we're very proud of it. We think we did a good job. This is the press release from the company, Neuralstem, who actually sponsored this trial, who I work closely with and I have a lot of respect for. But again, this is a press release. And the press release says this: secondary efficacy endpoints at 9 months post-surgery indicate a 47% response rate to the stem cell treatment as measured by either near zero slope of decline or positive slope of the ALS-FRS. We didn't say that in the paper, okay? And in fact, that may be true, but I don't have enough evidence to prove it. But the press release says that 47%, about half the patients, responded to the treatment. And I consider the term response as I got better or I had a positive effect. And I'm going to say it again, that may be true, but I don't have enough evidence and I don't have enough data to prove it. I'm going to do one more. And this is a very exciting, very important trial that's going on by a company called Brainstorm, okay? And they've now published one, and there's going to be some presentations at the meeting in the next few days from this company about a placebo-controlled trial, okay? They had 14 patients in this, what they call the phase 2A trial, and they looked at these patients. This is the ALS-FRS-R, and this is the forced vital capacity. And they argued here, which is kind of interesting, that the slope of decline that these patients had pre-treatment changed post-treatment. And you can see it here again. This is all the patients together, okay? This isn't any single patient. These are all the patients averaged together. And that's a very exciting, very hopeful thing. In fact, I'm going to go back one slide here to this. This is the reality of what patients look like. And so that's why you need so many patients in a clinical trial, because they're so variable. So looking at 14 patients, over this period of time and averaging them, I think is very hopeful, okay, and very interesting. But the press release says this: Israel's brainstorm says stem cell drug benefits patients with ALS. That may be true. I don't know that it's not true, okay? It may be true, but I'm arguing that we don't have enough data at this point to really say that, okay? So I think that's a little bit of hype. It's not the hype like the Chinese stem cells, but it's hype, okay? And so I think we have to be very careful when we read press releases. These things need to be tested in trials that are powered to actually give us a result that tells us whether it works or it doesn't work. So that's all I'm going to say. I'm done. I've written about this before, and if anybody wants to read about this, this is a publication that came out several years ago, and I go through these different arguments again. So thanks very much for listening, and I'll take any questions as well, or else we can wait until the panel discussion after. Yes, I'll take any questions. I'm sorry? We don't know if it slows the progression. That's the point. We don't know. We know it doesn't stop it. But we haven't done enough patients to actually see what happens. And there's such variability patient to patient that we have to be very careful that we have enough data in enough patients to really see what happens.
Sorry, can you just repeat the question?
The question was, go ahead, you can answer it. The question was, so stem cells slow the disease, but they don't stop it. Is that correct? Yeah. And my argument was, I don't know if it slows it. We have some interesting and very hopeful data that it might slow it. We have fairly good data that it doesn't make it worse, but we don't have data yet that says that it slows it down or stops it.
Hi. I wanted to ask if the outcome was analyzed with many factors like male, female, premenopausal, postmenopausal, because in our study of autologous bone marrow MNC intrathecal, we have found that premenopausal women who have a neuroprotective estrogen progesterone effect, they do the best among the group treated with the stem cell. So have you analyzed like a subgroup analysis?
I think the question was, have we looked at subgroup analysis of pre versus postmenopausal women?
Yeah, and male, female, age, onset of disease, because all these factors will affect the outcome.
Okay, so it's a good question. We have done that with our larger ALS population, and we published that as well. But in that, we published close to 1,000 people, and so we could actually have some statistical data. Personally, our trial only has 30 people, and in fact, we have very few women. And most of the women, in fact, if I think about it, all the women are postmenopausal, so I have no comparisons there. We do have an age range, but again, we don't have enough patients in this trial to actually see a difference based on age. Having said that, we've done that in the general ALS population, and turns out that age is a factor. The older you are, it seems that the more rapidly your ALS progresses, at least in our group. And there is an issue in women, although most of the women are postmenopausal that we have. But no, the answer is no, we haven't done that.
Dr. Glass, you mentioned that your work was being done with embryonic stem cells. I'm wondering if any of this work has been done with induced pluripotent stem cells.
We're not using embryonic stem cells. We're using what are called, they're really adult stem cells. They're fetal-derived stem cells. So embryonic stem cells come from what's called the blastocyst stage, where they can develop into anything. And in fact, an embryonic cell can make any kind of tissue. We're using cells that are a little bit more advanced in their development, and they come from the spinal cord of a fetus, from a single fetus, and those are fetal stem cells. This has not been done with induced pluripotent stem cells. That has not, in fact, I think a few years ago, we all thought that iPS cells were just going to be for research in the laboratory. That may happen someday, that iPS cells are used for treatment, but it hasn't started yet.
So we have a question from online. Would you please address the ethics of placebo surgeries with stem cells?
Oh, that's tomorrow's talk. I think it's a major issue, and there's a major controversy out there about placebo, especially if we're doing injections into the spinal cord. Injections into the spinal cord are dangerous, and they're expensive, and they're big-time surgeries. And so if you're going to open somebody up and stick a needle in their spinal cord and inject something, putting in something that's potentially non-active may be an ethical challenge. I agree with that. And so we need to come up with ideas for true placebo trials, or at least finding an alternate to placebo that can meet the scientific rigor that's necessary. And we have some ideas about that. We have some predictive models that may be able to work as a virtual control arm, and I'm happy to talk about that, but I don't have enough time today. But yes, it's a major ethical question, and I think that's for the next trials, it's going to be a problem. Jeremy wants to answer that as well.
I just wanted to make the comment that the ethics work both ways, because as John said, it's a major surgery to inject these stem cells, and there is morbidity, there are side effects. And so I think you would really, really want to be sure that such a therapy worked before you exposed a larger number of patients to receive these therapies. And so I agree with him completely that this is a very difficult question in terms of how do you conclusively decide whether a treatment works. But I think the ethics of providing a non-proven but dangerous therapy to people balances the ethics of potentially exposing people to danger without any hope of positive effect.
Sorry, is there anything being done to prevent it happening at all? All your work seems to be from people that have ALS. Like, is there, what work is being done to try and prevent it? People that have the gene, or
Right.
as inherit.
That is a great question to introduce my friend here, Jan Velding, because genetics is the way to do that. And so we don't know how to predict ALS. It's a relatively rare disease. It only occurs in about 1 in 100,000 people, and it occurs in all walks of life. So predicting ALS is really we need genetic markers. And the genetic studies are already underway for at least SOD1 and hopefully with C9orf72 in the next year or two. That's for people who are symptomatic. And if they seem to have some efficacy, we might start thinking about having people who are carrying the gene be treated to see if we can prevent it. But I'm going to let Jan talk about that.
One more time for questions.
Have you done anything research with PLS?
PLS, yes.
PLS for 19 years.
This lady's at PLS for 19 years.
Yeah. So PLS is an interesting story. If you think ALS is rare, PLS is pretty rare, more rare. And in fact, the first good clinical trial with PLS is now going on in the United States with a drug that's being supported by the Northeast ALS Consortium, which we're very proud of. But the idea of using stem cells for PLS is also a good one. In fact, I would argue that some of our patients had what I would call upper motor neuron predominant disease. I don't think they had pure PLS, but they had more PLS than ALS. And so we haven't excluded them from these types of trials. But there's been no trial that I know of with stem cells that are specifically for PLS.